Meiosis and genetic diversity. Table of contents. Meiosis process. Errors in meiosis. Meiosis. Mitosis is the process by which a cell divides, resulting in two daughter cells, each with the same number of identical chromosomes. It occurs to replace damaged or short lifetime cells and to support growth by increasing the amount of cells. Meiosis is where a sex cell divides twice, eventually creating 4 daughter cells, each with half the number of chromosomes of the original cell. Meiosis is key to sexual reproduction. Meiosis only occurs in germline or sex cells. Germline refers to the cells that pass on genetic information from parent to offspring. During the process, the genetic material from the original cell is scrambled, so none of the daughter cells have the same genetic material as the parent or each other. There are 2 types of sex cells, male and female. When they combine in the fertilization process, the chromosomes are further mixed. The combination of meiosis, fertilization, and mutations increase the genetic diversity of a species. The sex cells of organisms are called gametes. Eggs in females, sperm in males. In many eukaryotic organisms, the somatic cells, or non-sex cells, have 2 sets of chromosomes. They are diploid, or 2N. Gametes have 1 set of chromosomes. We call that haploid, or 1N, and they are produced by meiosis. Sexual life cycles alternate between haploid and diploid phases. Fusion of haploid gametes during fertilization results in a diploid offspring. Regardless of a human cell being a gamete or a somatic cell, it contains both autosomes and sex chromosomes. Human autosomes are chromosomes 1 through 22 that contain all the genetic information that makes an individual unique. Human sex chromosomes are the 23rd pair of chromosomes that determine an individual's sex. There are 2 types, X and Y. A female will have a pair of X chromosomes, XX. Males have 1 of each, XY. Other organisms have different numbers of autosomes and sex chromosomes. For example, fruit flies have 6 autosomes and 2 sex chromosomes. The adder's tongue fern has 1,440 chromosomes. The Atlas blue butterfly has 448 to 452 chromosomes. For humans, the male parent determines the sex of the offspring, since the female parent always contributes an X chromosome. If the male provides an X chromosome, the offspring is female. If a Y chromosome is provided, the offspring is male. For many birds, such as chickens, the female has different sex chromosomes, W and Z, so the females determine the sex of the offspring. Reptiles, such as sea turtles, have no sex chromosomes, but the temperature when the eggs develop determines the sex of the offspring. Sex determination is not chromosomal, it's epigenetic. A karyotype is a photographic inventory of a human's chromosomes. The chromosomes are digitally separated and ordered. Shown is a karyotype of a human male, showing 23 sets of homologous chromosomes with sex chromosomes X and Y, which you can see here. In order to view chromosomes in a karyotype, a metaphase chromosome spread is performed. Chromosomes are only visible at metaphase of mitosis or meiosis. So cell cultures are synchronized and halted at metaphase with a drug that inhibits spindle attachment or assembly. Cells are flattened on a sticky slide and stained to see the banding pattern, either with a Giemsa stain or a chromosome-specific fluorescent stain. Each chromosome has a characteristic length, centromere location, and banding pattern that allows us to distinguish it from other chromosomes. The technique is used to see large changes in chromosome structure, not point mutations such as insertions or deletions, but losses of parts of chromosomes, translocations, or the results of non-disjunction. Translocations are visible in a metaphase spread. A portion of one chromosome is added onto another chromosome, or chunks of chromosomes trade places. Meiosis non-disjunction events, which we'll discuss later, are also plainly visible in metaphase chromosome spreads. The pairs of matching chromosomes in the somatic cells of diploid organisms are homologous chromosomes. In humans, each somatic cell contains 46 chromosomes, which make up 23 homologous pairs. Homologous chromosomes share shape and genetic loci, each pair controlling the same inherited characteristics. One pair is inherited from the mother and one from the father. The sets are combined in the first cell following fertilization and then passed down by mitosis. Homologous chromosomes can carry different versions of the same gene. These versions are called alleles. Two examples are coat color and eye color in mice. Coat color, brown and white, are different phenotypes caused by different versions of the same gene or alleles. Eye color, black eyes and pink eyes, are different phenotypes caused by different versions of the same gene, alleles. The overall function of meiosis is to reduce the chromosome number from diploid to haploid, ensure that exactly each haploid cell has a complete set of chromosomes, and generate diversity among the products. Meiosis reduces chromosome numbers in diploid organisms to create sex cells. Like mitosis, meiosis is begun by a single duplication of chromosomes during the S phase. The meiotic cell cycle has 2 rounds of nuclear division and cytokinesis, while the mitotic cell cycle only has 1 round. Unlike mitosis, the overall result of meiosis is 4 daughter cells, each with half the number of chromosomes, haploid. The process involves 2 consecutive divisions called meiosis 1 and meiosis 2. In meiosis 1, the number of cells is doubled, but the number of chromosomes is not. So we finish with 2 daughter cells, each with 23 chromosomes. So we now have 2 haploid daughter cells. Meiosis 2 is a division process similar to mitosis. So we start with these 2 haploid daughter cells produced in meiosis 1, and we replicate them in the process similar to mitosis and end with 4 haploid cells. Prophase 1 in meiosis 1. The nuclear envelope disappears and the spindle begins to form. Homologous chromosomes replicated during interphase pair to form a tetrad. So we have 2 copies of maternal chromosome 5 and 2 copies of paternal chromosome 5. Crossing over occurs during prophase 1. Genetic material is exchanged by the homologous chromosomes when they line up together during prophase and form a synapsis or connection, joining together at a point called the chiasma, or plural chiasmata. So let's look what happened here. Capital A here picked up some genetic material from lowercase a in exchange for lowercase a receiving capital A genetic material. So we form this junction here, and we get this crossing over of genetic material. Crossing over at the chiasmata increases the genetic variation of the offspring. Since this can occur several times at variable locations in each tetrad, the variation that can occur between two parents is extremely large. This is one of the reasons that, with the exception of identical twins, everyone is a unique genetic entity. Metaphase 1. So we're still in the first phase of meiosis, meiosis 1, and at metaphase 1. So what happens here? The tetrads line up at the center of the cell, the metaphase plate. Each maternal chromosome could be on the left or right for each set of homologous chromosomes. This gives each cell a random combination of genes from each parent's chromosomes. This is called independent assortment. Let's jump ahead just a minute so we can see a little bit more about this independent assortment. Look at these two haploid daughter cells. Each gamete receives a haploid or 1N set of chromosomes that is made up of or comprises both maternal and paternal chromosomes. See how there's some red and some blue in these daughter cells? Along with crossing over, independent assortment accounts for most of the genetic variation in population. So the reason that this happens is because back here in metaphase, these chromosomes line up independently. So we can end up with both paternal and maternal genetic information in each gamete. Given n pairs of chromosomes, there are 2 to the n ways in which chromosomes can line up during metaphase 1. In humans, there are 2 to the 23rd or 8 million ways of combining homologs. This means combining human gametes can produce 64 trillion combinations in the zygote, just as a result of independent assortment. All right, let's go back now. The third step here in meiosis 1 is anaphase 1. The tetrad splits. Maternal and paternal inherited chromosomes move to one pole or the other, independent of the other chromosomes. And then the last 2 steps, telophase 1, the nuclear envelope reforms, and the nucleus is now haploid. And then interkinesis, which is the division of the cytoplasm, similar to cytokinesis. And at this point, the 2 cells are now haploid. The cells at this point go directly into meiosis 2. There is no interphase. Meiosis 2 follows the same steps as mitosis, with a few important differences. Meiosis 1 starts with 1 diploid cell and finished with 2 haploid cells. Meiosis 2 starts with the 2 haploid cells and finishes with 4 haploid cells. During prophase, meiosis 1 had crossing over, but meiosis 2 does not. In metaphase 1, tetrads or homologous pairs line up and centromeres do not split. In metaphase 2, individual replicated chromosomes line up and centromeres and sister chromatids split. Metaphase 2 looks a lot like mitosis, except there's only 1 copy of each chromosome. Here's a look at meiosis 2. In prophase 2, the nuclear envelope disappears and the spindle forms. In metaphase 2, the chromosomes with 2 sister chromatids line up at the equator. In anaphase 2, the chromosomes split apart, 1 chromatid moving to each pole. In telophase 2, the nuclear envelope reappears. And during cytokinesis, the cytoplasm divides, and we're left with 4 haploid daughter cells. Comparison of mitosis and meiosis. Mitosis starts with one cell and ends with two. It starts diploid and ends diploid. The process of mitosis goes through once. The homologs line up separately. There's no crossing over. The chromatids separate during anaphase, and it produces identical cells with identical complete DNA. Meiosis starts with one diploid cell and ends with 4 haploid cells. It goes through the steps twice. The homologs line up together as tetrads. Crossing over occurs. The chromatids only separate in anaphase 2, not anaphase 1, and it produces gametes that are genetically different with half of the required DNA.